Next, there, well, is that it? No.
There we go. All right. It's really a great privilege and an honor to be here to talk about Dr. Hal Broxmeyer's work. I was one of his last trainees. I joined his lab in early 2019, and we were still really in the thick of it when he passed away. It's really incredible how much of a profound impact he had on my career and on my life just in that short amount of time. And so I'm really grateful to be here to get to talk to you about his work. I titled this talk A Lifetime of Discovery, and I stole this title from this astronaut, Scott Kelly. He has this book subtitled A Year in Space, A Lifetime of Discovery, and I thought it was very fitting for Hal's career. Because when we think of Hal, we think of cord blood transplantation. We think of, the year in space for Hal was sort of that first cord blood transplant. But really, his entire career, his entire life was full of discovery, as evidenced by his many Lifetime Achievement Awards that I could list, but it would take my entire 20 minutes if I did so. Just a quick overview of what I'll talk about. I want to talk about some of Hal's historical work. I'll talk about his focus on cytokine and chemokine science, briefly about the first cord blood transplant, but Scott's going to really talk in depth about that in a little bit. And then his more recent work in improving umbilical cord blood transplantation. And I know a lot of you are probably curious about what he was working on in his last couple of years, I'll touch on some of that as well. Some quick important statistics for Hal. He had published almost 900 times during his life. He still has several publications that we're working on now and we're continuing to try and put out. It's been cited over 70,000 times, and he formally trained nearly 80 postdocs and grad students, and really informally mentored pretty much anybody who asked for help. He was involved in collaborations that were interdepartmental, international, you name it. And he was published in small journals like the New England Journal of Medicine and Science, Cell, and Nature. And really, if it was a reputable journal and he thought that the data was important, he was more than willing to put his work in and make sure that people saw the data, which I thought was really admirable. All right, so I'm going to first talk to you about some of Hal's work on cytokines and chemokines, which was one of his favorite things to work on throughout his career. And so some of his earliest work focused on just taking purified proteins, purified factors, and finding out what happens when you put these factors on cells. And I'm gonna summarize probably 30 years of work here in about 2 minutes, so I apologize if I'm being a little superficial about it. So one of his first areas of work were on these iron-binding proteins, lactoferrin and isoferritin. And what he found was that these iron-binding proteins, when put on hematopoietic stem and progenitor cells ex vivo, actually had an inhibitory effect on myelopoiesis. Really some basic scientific studies that I think laid the groundwork for studies that are still ongoing. And some of these iron-binding proteins now are actually being looked at as potential leukemia modulatory proteins. And so again, just some of the things that Hal worked on back in the 70s that are still very relevant and important today. He also did a lot of really comprehensive study on suppressive chemokines and cytokines. Again, basically just anything that was newly purified, he said, okay, let's put it on cells and see what happens. And I thought what was really cool about a lot of these studies, looking at a lot of these different proteins like the MIP family, IL-8, several different cytokines and chemokines, he really just reported the data in these studies. It was, here's what we put on the cells and here's what happened. And these ones had inhibitory effects and these ones didn't. And he always closed with, most likely all of these things, have to be taken into account that it's going to be different in vivo because all of these things are present together, not just one at a time, not just two at a time, and it's really dynamically regulated. And that was something he was still emphasizing to all of us at the end of his career, was that this was quite important for somebody eventually to be able to integrate all of this information and figure out when these things were actually doing what they were doing. And so he also was sort of on the ground level of a lot of the work with SDF1 or CXCL12 and its role in binding to CXCR4 and regulating the migration of hematopoietic stem and progenitor cells, of course, which is critical, we all know, for homing and for retention of these cells in the hematopoietic niche. And so when the initial studies were put out that showed this was a chemotactic agent, for myeloid progenitor cells, Hal did a lot of early work finding out whether it had other roles, like in combination with stem cell factor, whether it had another role in supporting the survival of these cells. And all of this work on chemokines really laid the groundwork for this seminal proof of principle study that Hal did, which was the first proof of principle study in a laboratory setting that AMD 3100 could enhance the mobilization of stem and progenitor cells to the peripheral blood. So AMD 3100, the CXCR4 antagonist that helped enhance the GCSF mobilization. And I'm stealing this line from Megan's eulogy of Hal that she gave several months ago. But we all think of the cord blood transplantation when we think of Hal, but he really played a huge role in the field of mobilized peripheral blood transplantation as well. So important things to keep in mind. So the rest of the talk will be focused on cord blood. Given where we're at, I think that's fitting, and given Hal's lifetime love of studying cord blood. And so I'm just going to briefly touch on his initial studies using cord blood, and then Scott's going to give you really some an in-depth look at it later on. When Hal first had these discussions about whether umbilical cord blood could be used as a transplantable source for transplantable hematopoietic stem or progenitor cells, what needed to happen was some sort of proof of principle. And so they acquired over 100 cord blood units, and he just did the ex vivo analyses that we knew at that time were the gold standard for testing hematopoietic stem and progenitor cell function. So they did colony-forming unit assays and found, of course, spoiler alert, that there were, in fact, promising stem and progenitor cells in these cord blood units, laying the groundwork, of course, for the case study, which is the first cord blood transplantation performed in France. And I would show some numbers or some data for this study, but Matthew Farrow's sitting in the front row. That's all the data you really need from this case study. And so obviously just a profound impact that this work had. And so for the rest of Hal's career, he really wanted to focus on how can we improve cord blood transplantation. So we know that one of the limiting factors of cord blood transplantation is the low number of stem cells. And so Hal's focus really became, how can we improve the collection, the expansion? How do we basically make this source as good of a source in numbers of stem cells? He believed overall it was a more promising source, but we needed to achieve that promise. And so I'll talk about sort of his four-pronged approach to that, which started with better collection and processing of the cord blood. So one of his newer focuses, his new baby, was studying stem cells in their normal physiologic oxygen tensions. So stem cells in vivo don't exist in 21% oxygen that we're all breathing right now, but in the bone marrow they actually exist around 3% oxygen. And even cord blood is much more hypoxic than ambient air is. And so Hal's work recently showed actually that isolating stem cells closer to that physiologic oxygen tension preserves functional stem cell numbers, and really laid the groundwork for using antioxidants and other molecules to sort of mimic this to potentially enhance the collection of functional stem cells for cord blood transplantation. He also did a lot of work on how to improve the homing of hematopoietic stem and progenitor cells used for transplantation. And one of the things that I'll focus on here is the DPP4 inhibition, because it was something that really showed a lot of promise. So some of the original studies on this were done by Kent Christofferson in the early 2000s. showing that inhibiting DPP-4 could improve the homing of the stem and progenitor cells to the bone marrow. There's actually been clinical trials for this as well, a couple of clinical trials for it, and so it's a really promising new mode of treatment. He also did a lot of work on stem cell expansion ex vivo, for instance, using this protein DEC. This is Megan Capitano's baby that Hal helped her get started, that she's now following up on in her lab, which is kind of demonstrative of Hal's enduring legacy, that work is going to continue. And so you could treat cells with this DEC protein ex vivo and get an expansion of functional stem cells. And I keep saying functional stem cells because Hal had it hammered so hard into my head that we don't just want to improve stem cell numbers by immunophenotype or anything like that, but we actually need to improve their functionality. You have to have a better transplantable unit. And so if I repeat that over and over again, it's because it's just hammered into my head by Hal. And of course, there's some crosstalk here. We can enhance homing of the expanded stem cells, and then of course we have other things that were explored that improved engraftment, maybe by unknown mechanisms, but things that were also just improve the engraftment overall. And so now I'll take the last couple of minutes just to discuss some of Hal's most recent work, some of the stuff that we were working on with him. His passion for scientific discovery really never let up. So in 2021, he had almost as many publications as he'd ever had in any other year, actually the most productive year since 1999. So he really just kept pushing forward and kept driving his science forward. And so the last few years, he really continued his work on chemokines. As I mentioned, that DEC chemokine that Megan studies. This was the most recent study that Hal and I put together. The last email I got from Hal was one-word email, submit, period, and that was for this paper. And so kind of a special paper for me, but basically what we found was that leukemia inhibitory factor can be cleaved by DPP-4, and that leukemia inhibitory factor also has a pro-survival effect on hematopoietic progenitor cells. By blocking DPP-4, we can enhance that effect. And so again, he loved working on chemokines, and so we just kept working on them. He continued his work on hypoxia. He showed that you could actually enhance the collection of mobilized peripheral blood stem cells by isolating in hypoxia. Showed some work with T-cell development ex vivo in these hypoxia studies, and really a lot of other studies. This was really the main focus of his lab the last couple of years. And of course, he kept his eye on the ball and really wanted to just make sure that we could identify or improve functional stem cell numbers. And so I'll just highlight this study by one of his more recent grad students, Tao Trin, who showed that leptin receptor, a cell surface protein, could actually be a marker for highly functional long-term stem cells. So really just continuing to contribute to that field of functional stem cells right up until the end. And he really just let us work on whatever we wanted to work on. As long as it was somewhat in his purview, if we had an idea, he let us follow it through. And so during the pandemic, when we were all kind of searching for something to do, he let us look at this study and where we were able to explore the effects of spike protein, the SARS-CoV-2 spike protein on stem and progenitor cells, and found that it did, in fact, have a profound impact on their function. And so the last couple of slides here, I just want to talk about Hal's legacy. His scientific legacy obviously lives on. This was a paper that was published about a year before Hal passed, and I think we're going to see a slew of these papers with the DPP-4 inhibition. This one shows that it can help prevent acute graft versus host disease during transplantation. He has another clinical study that shows that it helps early recovery as well of cord blood transplantation. And Sharif, who's been his collaborator on that, I think is just going to continue to push these papers out. He also has a couple of studies that came out recently from his most recent grad student, Arafat Al-Jufi, who showed that TET2 is actually downstream of the hypoxia signaling. So all this hypoxia work that we've been doing, he started really digging into the mechanism here and showed that TET2 is downstream of that, of those effects. And another study by one of his other recent postdocs, Alex Wang, who showed that TFE3 nuclear localization is regulated by changes in oxygen tension, and that it could have a profound effect on hematopoietic stem cell transplantation. He did this in human cord blood cells, which is really interesting. We hadn't yet had a really good way of testing the hypoxia with the cord blood cells, so it's a promising step forward for that part of the field as well. So this, in Hal's never-ending effort to see how long cord blood cells could be preserved for. And there's something that he emphasized, that we really need to know how long these cells are good for. Are they good indefinitely? Or do we have to put an expiration date on them? He sought out, before he passed, he sought out these 3 samples from New York Blood Center that were 27-year-old cryopreserved cord blood units. And so he passed away before we were able to start this project, but Scott Cooper and I decided to continue on with it, as a tribute to Hal. And so really just asking the question, are these still functional cord blood units? And what we found pretty remarkably, actually, was that we have highly engraftable hematopoietic stem cells in these 27-year cryopreserved cord blood units. And to us, it suggests that age itself should not be an exclusionary criteria for these cryopreserved units, which I think is really important for the field. And we were also able to harness this data. We had 2 units that engrafted really well, very highly functional, 1 unit that did not engraft nearly as well. We were able to harness that actually and do some molecular profiling to get some hints at what is different between these units. So regardless of whether it's because of the cryopreservation, the age of the unit, or just an inherent property of the cord blood unit itself, why is one unit better than the other? And we actually were able to identify some genes and gene programs that are enriched in those highly engrafting stem cells compared to the low engrafters. And we think this really lays the groundwork for a lot of really important studies for the future. And so Hal's scientific legacy certainly lives on there. But those scientific legacies, while important, I think don't match up to really what his legacies are. This is Sophie Lee. She was diagnosed with MDS when she was 5 years old. There were no matches in the bone marrow or peripheral blood registries, so she got a cord blood transplant instead. And Sophie Lee is now a happy and healthy 12-year-old girl. And I think that's really Hal's legacy that lives on. Tens of thousands of people whose lives have been positively impacted by Hal. And so he was a real-life superhero. It was a true honor to get to work with him, and I thank you for your time.
We're going to hold questions till the end, but thank you, Jim. And I now invite Megan Capitano to speak. I heard Megan speak during a memorial, I guess, session for Hal that Indiana University held, and it was a Zoom session because of COVID. But I was just so impressed with her, how eloquent she was about describing Hal as a mentor. And I know, of course, she had personal experience for that, but I wanted her to talk about that with you all today. So, Megan, thank you.
I'm really grateful that I got asked to speak about Hal today, and I apologize because still when I think of Hal, I get a little teary-eyed, so I'll try not to. But I was really lucky that about 11 years ago, I had, when I was getting my PhD, that I had the honor of having, ooh, why is this moving on its own?
Oh, I'm sorry, but left is forward.
All right.
Right is back.
Perfect, thanks.
You must have touched that.
Yeah. I was really lucky to have a mentor, Dr. Elizabeth Rapaski, that really believed that if there was somebody coming to speak that would be of interest to us as students to meet, she always gave us the opportunity to meet with them. And it just so happened that Hal Broxmeyer was coming to give medical grand rounds at Roswell Park Cancer Institute. And she's like, well, I'm meeting with him. I have a half-an-hour meeting. You should join me because you're interested in hematopoiesis. So why don't you sit down and talk to him? So she pulls me into her office. She's like, this is Hal. Hal, this is Megan. She wants to tell you a little bit about the project she's working on. And it turned out that that project I had submitted to Blood, and Hal was reviewer too, and I didn't know that at the time. And he's like, oh, I know about your project. You want to do a postdoc in my lab? 5 minutes into the meeting, because he had read the paper and he really liked my story ideas. And so I interviewed on the phone and talked to him about a postdoc position, and he pulled me, he spoke with so much enthusiasm about everything he did. So like, it's hard not to say yes to working with somebody who just so obviously loved what he did. And so I was like, oh yeah, I'll come do a postdoc with you. And so he invited me to come up to Indianapolis to get to know his lab people. And I was so nervous because he's such a big name in the field and I'm just a graduate student at the time. So I was like, oh, and he goes, I could tell you're so nervous. Why are you so nervous? I already offered you the job. It's yours, so you shouldn't be so nervous. But it was one of the greatest honors and best opportunities of my life that he provided with me in that 5-minute meeting I had with him. And one thing Hal will tell you is your future is all dependent in science on not just how hard you work, but who you surround yourself with during your career. And part of that is by picking the best mentor for you. And one thing I absolutely adored about Hal is that he loved talking about the history of his field. He loved talking about his experiences over the years. And he talked about how his mentors shaped his career. So Jim talked about Hal's passion for studying cytokines and chemokines and how that regulates hematopoiesis. And a lot of that stemmed from Hal's mentors. And one of those mentors was one Hal started as a research associate at Sloan Kettering and then moved on to his first faculty position. And that was with Malcolm Moore, who, as many of you guys know, is the father of GCSF, if you call Hal the father of cord blood. So Malcolm is the father of GCSF. And so Hal could, and Malcolm would tell you, and I've had the lucky opportunity of meeting Malcolm several times, tell you all about how GCSF was discovered. And they would go on and talking about how that led to multiple different therapeutics for stem cell biology and neutropenia in the clinic. But Hal's career was most definitely driven by his interactions with his mentor. And he made sure to take the lessons he learned from those mentors and to apply it to his own mentees. And so part of the mentor process, Hal would tell you, is not just the mentorship with somebody who's more senior over you, but also with your collaborators and with your colleagues. And so through his beginning of transition from research associate to faculty position, he was surrounded by others who were also moving into faculty positions. And one of those people was Richard O'Reilly at Memorial Sloan Kettering. They moved up into their first faculty position. They had their first major promotion at Sloan Kettering around the same time. And as you know, many of you might know, Richard O'Reilly went on to be the head of pediatrics at Sloan Kettering and ran their bone marrow transplantation program there for a long period of time. And when he was stepping down from the head of pediatrics, Hal and myself went up to Sloan Kettering for a symposium that Memorial Sloan Kettering was having for Richard O'Reilly. We got to sit there and have dinner with Malcolm Moore, Richard O'Reilly, and Beau DuPont. It's another big name in the transplantation field. And just listen to all of the history that all these men did together, which was a highlight of my career. So during Hal's time, he got to develop his first lab at Sloan Kettering, his own first private lab at Sloan Kettering. And during that time, he got to invite several mentees to join him in that lab. And I'm not going to go through all the names, but I want you to be aware, this is what Hal wrote about each of his mentees. He collected where they came from, what they did in his lab, and where they went for every single one of his formal mentees. He kept in track of everybody's career. He kept in touch with everybody. He did not forget you. He would never let you flounder on your own. He was with you your whole career. So I want to just point out there will be a couple names you'll see on the list throughout his career that will pop up again and again, because as I said, he doesn't forget. People that had been in his lab when he was at Sloan Kettering will pop up and do their sabbaticals in Hal's lab when he goes to IU. So Hal gets recruited away from Sloan Kettering early in his faculty career by Ron Hoffman to come to join Indiana University School of Medicine in 1983, where he stayed for the rest of his career. Here's some badges. You can see Hal's career spanning an IU just from the change in the badge format. But Hal started his career at IU in '83, and you know this is when a lot of that initial cord blood work, even though the ideas started with some collaborators in New York that continued and really thrived in his time when he was doing his own research at IU. But while Hal was at IU and had his mentees in his lab, and I'll go through that in a bit, Hal was also the head of the Walther Oncology Center for a good portion of that time and chaired the Department of Microbiology and Immunology. Hal's mentoring was not, as I said, not just the people under him, but the people surrounding him. He mentored faculty, he mentored even people in business and helped them drive different areas at IU. So he really started and helped start this, like, the cancer center's more organized research facility. And it started off as the Walther Oncology Center. I have to say, a couple years ago, IU became a comprehensive cancer center, in a large part by Hal's passion to drive that community to band together at IU. So through the years, you can see, and this is also what's amazing about Hal, is the loyalty of the people surrounding him. You look at the pictures, and it's going to be hard to see, but a lot of those people stuck around with him their whole careers. And actually, you can see one of those people sitting right there, Scott Cooper, who, Hal inspired dedication. And Scott's an example of that. And another example of that is Mark Kaplan at IU School of Medicine. Hal hired Mark Kaplan for one of his first faculty positions in 1997. And Mark is now my boss, the head of my department that I'm an assistant professor in. And so I'd asked him, what was it like for you to have Hal as a mentor? And he goes, Hal was the primary reason I came to Indiana University. He was the newly installed chair of microbiology and immunology, as well as the director of the Walther Oncology Center. While a junior faculty member in the department, Hal provided mentoring in a variety of ways. He reviewed grants and offered advice, but perhaps most importantly, he offered support in being confident in my success. And perhaps that was what always made Hal a strong mentor. He was demonstrative with his confidence in his faculty and his trainees. He expected you to be outstanding and successful. It was that simple. He provided support and guidance, but your success was your own. As chair, I can see the continued impact Hal has in the department, in the cancer center, and in the scientific community at large. In many discussions, Hal would have the insight to reiterate the goals that are important. As he might say, the rest is of just crap, although he probably used more colorful language, which is true. He probably did. And actually, that initial quote did have more colorful language, but Mark wanted to clean it up for the presentation. But Mark went on and told me that he didn't really think he'd make a good chair of the department. He was not very confident in his ability to do it. But Hal pushed. I was like, of course you have everything you need to be the chair of the department. What are you talking about? Have more confidence in yourself. He did that to me too. I worked for Hal for 4 years as a postdoc and 4 years an assistant, 4.5 years as an assistant research professor in his lab. I probably would have been happy working as Hal's assistant research professor for as long as he would have let me, right? Because I loved working with Hal. It was one of the best experiences, if not the best experience in my scientific life. But Hal's like, you have everything it takes to have your be your own PI and run your own lab. And he gave me that confidence. So I applied, and I applied for one position, and I got that position. And I got to work in the same department with Hal for a year before he passed as a assistant professor, tenure track professor. And now I'm actually in Hal's lab space continuing his hypoxia work. We have a core facility that we were running together before he passed, part of the Cooperative Center for Excellence in Hematology at IU, studying hypoxia. And I wouldn't have been able to do that without Hal's encouragement either. But as you can see, I'm not going to read through these names because Hal mentored a lot of people. But there is one person in particular, as I said, will pop up. Patrick Gentile, MD, he had been Hal's mentee at Sloan Kettering, popped up and did another year of sabbatical with Hal during his career. Out of all these mentees, and they're extensive, Hal followed them throughout their career, never stopped having contact unless they chose not to keep in contact with him. He knew where all of them went. He knew where their careers were going and was happy to help them if they ever wanted it in the future. He never turned down an opportunity to help. Hal would give you the shirt off his back if he thought you had promise. Hal literally would give you all the resources you needed. He was an excellent mentor, especially for postdocs and junior faculty because of that. Because if he believed in you, he's like, well, I got some money. You want to do that experiment? Okay, let's do it. You don't have the grant money for that? Oh, you need a little bit more lab space. You need some equipment to use? Come into my lab. Do it. He never, ever would turn down an idea if you were passionate about it and there was a rationale behind why you wanted to do it that he believed in. He always gave you an opportunity to do whatever research he thought could drive the field forward. So if I keep going through here, he mentored MDs, PhDs, students from different countries that just wanted to learn a technique that didn't have the opportunity. We actually had a student come in and spend a year in our lab from Mexico because she wanted to learn some of the techniques our lab was doing. So Hal invited her in. Scott and I took her under our wing and we showed her all the techniques she wanted to learn. And now we're getting into his more recent. In red are the people that were in his lab when he passed. Tao finished her PhD before Hal passed. Arafat did it a few months after. Large thanks to Jim and Scott for helping him get through that. Alex and Jim are continuing to work on projects that they had started in Hal's lab. Alex is doing this now in Ruben Kapoor's lab at IU, and Jim's doing it with assistance from Mark Kaplan and myself. And what's so great about this is the department is still very supportive of Hal. They're letting that work continue. They're giving the money to continue that research so that Hal's legacy can move forward. Out of overall, formerly Hal had 76 total trainees and mentors. out of those 76 that came in as junior people, so either graduate students or medical fellows or postdocs, over 35 of them have their own labs now. And out of that, or running a clinical division. And so you can imagine how his career has moved others' careers. Imagine how many people those individuals have mentored. How the big, wide effect that Hal's career has had on the scientific community. That's just who he had in his lab. Hal was active in many organizations as well. And Hal was a very passionate and active participant in the TRTH program, which is the Translational Research Training in Hematology that's co-sponsored by both ASH and EHA. And one of those people Hal had mentored through this program is Fabiana Perna, who was an assistant research professor at Sloan Kettering in 2011 when she was being mentored by Hal. She wanted to say to Hal, "Thank you, Dr. Broxmeyer. May you rest in peace. Years of honest, wise, and inspiring mentorship. I have no words to describe how much you meant to me over the past 10 years. Hal and Fabiana never, as I said, he kept in touch with his mentees. They never lost touch. And in fact, Hal was able to recruit her into an associate professor position, and she's now also at IU School of Medicine. And she and I are actually close collaborators. My very first grant as a faculty mentor was with Fabiana. And then finally, just to finish with my experiences with Hal as a mentor. As I said, Hal was the perfect mentor for postdocs. You had an idea, you pitched it to him. He wanted short elevator pitch. Unfortunately, I am not known to be brief in anything. I wrote him 6 pages of pitch with figures and references. And he would go, "Well, no, don't go into that area. You don't want to be in that, no." Or like, "Oh no, that's a great idea. Let's do it. But you want to do it in a more clinical relevant. Let's do it with cord blood instead of mice." Or in other areas like this, Hal would be like, "Let's go." If he liked your idea, he wanted you to move on it right away. So you don't wait around a day, you did it the next day. And he gave you all the money you wanted to do the projects with, all the supplies. You need this protein? Oh, I know this guy who makes this protein. Let's talk to him. Or like call him right then. He didn't wait. He pushed. He was aggressive in getting the science out, which as a mentee sometimes was a little overwhelming, but it was good. Hal and I together in our formerly 8.5 years working together, but my 10, 11 years mentorship with him, we have over 36 publications together. I think it got to the point where we worked so much together that we started dressing alike because this was Hal's presidential medal ceremony, and they had asked former mentees to come and talk about their experiences with Hal. I was picked to be his current person to give a talk about their experience with Hal, and we came wearing pretty much the same outfit: pink shirt, black pants. Hal was a firm believer. He loved going to conference. Oh, he loved listening to everybody's stories. In fact, you could pick Hal out, even though he was not a very tall man. You could find him at any conference because he always wore a nice suit, but white sneakers. So you follow those bright white sneakers while he sped walk throughout the meeting. But when he went to meetings, he didn't want us to just think about science the whole time. He wanted us to enjoy the place that we went. So we're a big participant at IU in the Midwest Blood Club when they were actively having their meetings. They haven't had one since COVID, but I'm hoping they'll get those back up. We were in Memphis, and he took the whole lab he was a huge Elvis fan, so he took the whole lab to go see Graceland and took pictures and bought memorabilia, and I actually have some of that Elvis memorabilia now in my office, including a bobblehead of Elvis. And then I said, Hal was a lover of scientific history. It wasn't just, though, in hematology or biology. He pushed science of all fields, physics. He was a huge Albert Einstein fan. So even up to one of our very last, actually, I think this might have been my last meeting with Hal. He's pushing books onto Jim and I that we should be reading. So this is our Zoom with Hal as he's showing us, he's just holding up books that he wants us to buy and read that he thought were interesting. Hal and I sort of had an informal book club going. Where you'd come in the morning and there'd just be a pile of books with a Post-it note, read these, you'll like them. So I'm trying to continue what Hal taught me as a mentor, the give everybody a chance. If the idea is good, push it, let them go. I don't have the resources necessarily to be able to do it as well as Hal does. I don't have the endless supply of money. I've only been a faculty member independently for 1.5 years, so I'm still working on that part. But if my students have an idea, I try to let them do it. If Jim wants space in the lab, let him have it. He is a brilliant man and is going to go far in his career too. So taking what I learned from Hal is surround yourself with people that you like to work with, and surround yourself with people that are passionate. You don't have to be the smartest person in the room, although Hal probably was. But if you love what you do, and you're passionate about it, and you work hard, you're going to be able to move your career forward. Because to Hal, that meant more than your book smarts. Because of the relationships I built with Hal, he introduced me to all the people he mentored and collaborated with, and I still find myself working with those people. So Fabiana Pernas, you see the person that Hal mentored through TRTH, and I are still collaborating together. We hosted our first session together at ASH last year. We got to have a grant together, had a grant together, just finished. And I'm continuing on mentoring in Hal's lab space with Jim in the lab, which was a Hal mentee, and Decal, which was a Hal mentee that I've taken on so she could finish her graduate school. With that, this is just a thank you from Hal's former mentees in various links. There is to say, Hal mentored people from around the world. So this is Alex, who's from China, thanking Hal in Chinese. This is Tao, originally from Vietnam, thanking Hal Vietnamese, Arafat in Arabic, Dung Yup in Korean, and Jim, because he thought English would be boring, did it in C++ computer programming. So Hal's mentees can never thank him enough. Anybody who's worked with him could never, or as a collaborator with him, really I don't think could ever thank him enough. He touched all of our lives with his passion, and that passion made you passionate. So I can't thank him enough. And that's it.
Thank you, Megan. Our next speaker is Scott Cooper, who was with Hal from the beginning and is going to tell the story of the first transplant and probably a little bit more. So thank you, Scott.
So, good morning. Good morning. Thank you very much for inviting us. Is this? Oh.
Front and back.
Front, back, okay.
Forward, back.
I want to thank you, Joanna, for inviting us to be here. It means a lot. It's been a tough year in the lab. A lot of things have changed, with Hal gone, but there's not a day that goes by that we don't think about him. So I've been asked to share the story of the first transplant. I've been with Hal since he came here, which is a long, long time. It's 39 years. So my story may be a little different. It's not going to have much data in it. Joanne used the word experience earlier, so I'm going to tell you a lot about my experience with Hal and with that first transplant, and some of the things that led up to that. I think the ideas and the steps that kind of led to that first one, Matthew. So there he is. I love that man. He was a good friend. So, we'll start with some of who Hal was before he came to Indiana. He was born in New York, Brooklyn area actually, spent most of his young adult life there, was very avid in athletics, loved track and field. And he was on the track and field team in high school, and when he went to college, in that lower right-hand panel, you can see he's chucking a javelin. And I say that because one day we were in his office, and we're talking about experiments that have to happen over the next few weeks, and the FedEx guy walks in with what looked like a big poster tube, about 5 feet long. And he stopped the meeting, and he got very excited, and he stood up. And you can imagine what was in the tube. He'd ordered himself a javelin. He talked favorably about his past all the time. His track and field days in college, he encouraged his kids to run in track and field in their high school. But he opened this thing up and put it together, and I said, what are you going to do with that? You know, there's no place to throw it. And he said, I'll find some place to throw it. But he loved his past. He loved everything he did in his past. He talked about trading baseball cards in the streets of Brooklyn with his friends. And when he passed away, he had a baseball card collection that was probably very valuable. Thousands and thousands of cards that I think Beth had to figure out how to get rid of. After completing his after completing his undergrad degree, it was a problem for him because he loved competing, but there were no post-grad track teams that he could participate on.
We're having a little trouble advancing
Yeah.
the slide.
Oh, we'll go the other way. Okay, there we go. So he had to figure out it's not going forward. He had to figure out some way to channel his competitive nature. We can go back, but we can't go forward. Oh, there you go. So he took up weightlifting, competitive weightlifting, not just, standard run of the mill, go to the Y and lift some weights. He did it to compete. And he didn't do much of anything unless he could either succeed at it or have some level of success or win. And you would think that this might end with the rigors of, you now have a position at Sloan Kettering, and then you have you've been recruited to a new university, and you're starting your own lab. And then you're appointed the director of the Walter Oncology Center, and then the chairman of the micro department. But no, it continued on. Again, it's not going. Next slide. There we go. Into his 50s, until he had hip replacement surgery. We tried to get him to wear these tights in the lab a couple times, and he wouldn't do it. But I think this competition was the Masters World Weightlifting Championships, and I think he won this one. Of course, he would tell you there were only 2 people in his weight category, so it was easy to either come in first or second. But the thing about this is that he always pushed himself. He never just relaxed, and for what it's worth, you could say that's not good, but it was kind of who he was. He never did anything halfway. He was always pushing, and always trying to win. So now we'll go back to the science a little bit. Next slide, please. So after getting his undergrad degree from Brooklyn College, he went on to get a master's degree from Long Island University and his PhD from New York University in experimental hematology, which in the 70s was a pretty new field. And then he did a postdoc at Queen's University in Ontario, also in experimental hematology. And after he took a position at Memorial Sloan Kettering, after that he took a research associate position at Memorial Sloan Kettering Cancer Center in New York. And here are just a few pictures of him in those early days as a real-life scientist. And I want you to pay attention to this microscope that the microscope that he's looking at in both those right-hand panels there. He stole some stuff from Sloan Kettering. Yeah, we'll get to that slide in a minute. I think he's beyond prosecution at this point. So when he came to Indiana in 1983, I had a position. I had taken a position with the IU Med Center right out of finishing my undergrad degree in an infectious disease lab, isolating chlamydia from genital warts. And so I was looking to change my career path. So he had this tech position open. I went and interviewed with him. And I had never met anyone as tenacious and focused and intimidating as him. You would think that the world was going to come to an end or the sun was going to fall out of the sky if he didn't accomplish what he was going to do. I'd never heard of hematology before, but I knew it was better than genital warts. So I took that position with him and never looked back. It was he was a great boss from the beginning. He was pushed and challenged and taught me a great deal. Next slide, please. So when you come to the university, they take some promo pictures, they put in their bulletins and this and that. And so this was Hal's, maybe first couple months at IU. And this is actually just a fun little quiz slide. One of these pictures doesn't belong. The picture is him with technology. Hal wasn't really a friend of technology, and if you know him, he handwrote everything. Every manuscript he ever submitted was always handwritten, and then given to his assistant, and she would type it out. Every experimental protocol was handwritten. I have notebooks of historical documents that Hal handwrote, and it was a struggle to get him to learn how to use email, but he did. But that was about as close to technology as he actually came. But sometimes I think my life gets locked up if I can't get my phone to work or I ran out of batteries. He never let that happen. Even if his phone died, he would just continue on, and because that's just the way he was. All right. So now we'll go back to the cord blood story, which is what I was asked to do. Next slide, please. All right, next slide. So while Hal was at Sloan Kettering, he made many friendships and many collaborations. And one of these friendships was with a guy named Ted Boyce. I think Ted and Judith were a couple. And he told me that, in their his many conversations with Ted, sitting around talking about hematopoiesis and hematopoietic stem cells and where they come from and what happens, he opined that, during this pilgrimage from fetal liver to the bone marrow, and they're floating around in the blood, could we collect them and do something with them? And I think that was, at that point, that idea of making this material functional and utilitarian was born. So some other connections that Hal had while he was in New York, next slide, please, was that he knew Arlene Auerbach, who was a physician scientist at the Rockefeller University. And Arlene was one of the world's leading authorities in Fanconi anemia. At that time, Arlene's lab was the only lab in the country that did diagnostic testing for Fanconi anemia. And in 1982, She founded the International Fanconi Anemia Registry, which was intended to provide researchers who study Fanconi anemia access to a large number of patients and diverse features of this rare disease. So during his conversations in relationship with Arlene, they discussed the possibility of collecting cord blood and using it to correct the hematological maladies that accompanied some of the Fanconi patients. And so they thought it was a good idea. So because of Arlene's database and her ability to identify patients with Fanconi anemia who had mothers who were pregnant with an expectant child, they could possibly identify a recipient and do in utero tissue matching and find out whether that newborn was going to be a good match and go forward. So then the search was on. So next slide, please. So enter Joanne Kurtzberg. Around 1985, Joanne had a young patient by the name of Matthew Farrow, who'd been diagnosed with Fanconi anemia and whose mother was expecting another child. I think Joanne knew Arlene through the diagnostic process or some other professional connections. And Arlene reached out to Joanne about this cord blood idea, and Joanne was very receptive about harvesting and using the cord blood from Matthew's new sister. Okay. And so conversations ensued, trying to determine, how we actually go about, when we go about collecting, and how we'd go about transporting, et cetera. And so while those conversations were going on, all the wheels were starting to turn. Hal knew that there were some basic questions we needed to answer in the lab about cord blood, just to make sure we didn't overlook anything. So another collaboration. Whoop.
Not working.
Not working. Next slide, please. Okay, so some of the questions that we needed, I'll just point to you for the next one. Questions we needed to answer were, how do we get samples shipped? Do we need to ship them on ice? Could we just ship them at ambient temperature? Hoping that this was going to really take off. It could be, hundreds, thousands of samples. They could be very expensive. So plus if you needed to keep it cold, it was important to preserve the HPC and the HSC. How long did we have, next line please, how long did we have before viability of progenitor numbers started to wane? If it got left on a dock somewhere, we wanted to know if you could still use it if it was 3 days old. How many HPC and HSC were contained in cord blood? And was there much variation? I say HSC, we'll come back to that in a minute. Were the HSC comparable to the bone marrow itself? And would the cryogenic process and freeze thaw kill too many of those progenitor and stem cells to make it usable? Because unlike a scheduled bone marrow where you know the day you're going to do the harvest and you're going to do the infusion, you didn't really know what was going to happen with cord blood. When the baby was going to be born, you didn't want to precondition your patient and have them sitting there waiting for the baby to be born. Plus, I think in the early ones, the desire was to have the donor sibling old enough and large enough that they could act as a backup in case the cord blood didn't take. So we had to cryopreserve everything. So let's see if it works. Next slide, please. So another relationship that Al had was with Gordon Douglas, who was the head of obstetrics and gynecology at New York University. And so Hal approached him and asked him if he'd want to collaborate on this project, and he said absolutely. So over the course of the next few years, Gordon shipped well over 100 cord blood samples to us. And in those days, it wasn't as difficult as it was now. There was no informed consent. It was basically, a sample that outside of a few clinical parameters you might want to test for, you just take it, and it really was a trash material. So sending trash in the mail, draining the blood out of it and sending that wasn't a big issue. And I don't think IRBs were really, I say this because it's difficult for us, even at Indiana University, to get samples to work with. And so we have collaborations with a few people in this room to get samples to work with. So these samples would come in, and we evaluated them for, the only stats we could take on them were volume, nucleated cell counts, HBC content, via colony assays. It's an agar assay and a methylcellulose assay. And then we would freeze them and thaw them out a couple months later, and we collected data on all hundreds of these samples. And for all you young kids in the room, back in the 80s, I mean, you may ask why we didn't do, long-term, short-term, HSC, MPP, CNP. There was no flow. There were no antibodies. There were no antigens to define a stem cell or even a progenitor cell. So the only way to really get a readout for a potentially engraftable progenitor cell or stem cell was to do a colony assay, and it was that last little compartment, the CFU GEMM. That was the closest readout we had for an engraftable cell. Plus, we didn't have recombinant factors, so we made our own conditioned media from a bladder carcinoma cell line 5637, and that contained all the cytokines we needed. It's kind of, we did have electricity, so. So, next slide, please. So then the hunt was on. They were, Arlene was hunting for, potential transplantable candidates. And I think the first one that was identified was the Curry family, Natalie Curry, who had Fanconi anemia. And her sister Emily was expecting. So they collected that blood. That was the first sample we stored in the lab, but it was the second one that Ilian transplanted in Paris, at least from our end. And then soon after that, Matthew's sister Dorothy was born, and we collected that and froze that in the lab. And just to let you know how early this was and how desperate we were to make this work, and we really wanted to have kind of proof of principle here. Dennis English was an assistant, I think he was an assistant professor in Ted Gabig's lab, HEMONC, and he actually had a connection with a brand new stem cell lab. They had a programmed freezer, program rate freezer. And so he's the one that actually did the processing and the freezing, and I did all the culture analysis on it. But we didn't have any infrastructure to, I told you I was going to tell you stories, to protect these bags. So we ended up taking these frozen blood bags, tying a string around them, and dropping them down in our liquid nitrogen tanks. And that's where they stayed until we used them. You make do with what you have, I guess. So this next slide, please. Oh, there we go. So this is actually Matthew's data. This is the setup sheet on the far left, and then the fresh cord blood. I was gonna bring this and give that to you. You can put it in an album. And then those are the actual colony counts. You can see at the bottom we set it up plus minus Heman. We didn't know if Heman actually was a good thing or a bad thing at that point, but in culture we just set it up every way we possibly could to get as much information as we could. We set it up at 3 different cell concentrations in 2 different assays, and then a couple months before transplant we'd thaw it out, reevaluate it. So just a little bit of history there. Next slide, please. So somebody had to score all those plates, and that was Hal. This was Hal's happy place. Hal had his best thoughts, his most creative ideas, the ideas that tormented a lot of us. He'd come up with experiments that, he'd hand me a sheet and say, I need this set up on Thursday, and it was 600 plates. My only consolation was knowing that he had to score them. And I never understood how he could score so many plates until I walked in on him one day, and next slide, please. Realized he had magic counting glasses, which he could just rip through those plates as fast as he could. So the decision was made that it was go time. It was transplant time. And I'm not really sure how that decision was made, whether it was a declining physical condition for Matthew, or whether the sibling, Dorothy was old enough and big enough, but the decision was made, we're transplanting Matthew Farrow in September. So Hal came to me and said, it's sometime in the summer of, '88, and said, you need to start making plans to get the cord blood from here to Paris, because they identified Ilian as going to be doing the transplant. She was an expert in Fanconi transplants. So we had to buy a shipper. We had to get FAA exemption to get liquid nitrogen in the passenger compartment, because nobody wanted us to check this as luggage. They didn't want it to leave and he said, you're going to take it too. So he said, we don't want it to leave your sight. Once you keep hands on it the whole time, it really had to work. So we bought this great big shipper. It weighed about 70 pounds. It had a roller thing on it, and it was the only dry shipper available at that time. But through the next couple months, I played phone tag with the FAA. Never got the same person trying to get an exemption so we could take liquid nitrogen inside the compartment, passenger compartment. They finally came through with a document. Then we had to clear it with TWA. And then the last hurdle was just getting it cleared, letting Indianapolis International and Kennedy, the stopover, and then Charles de Gaulle, those airport authorities let them know what was coming through. Because when you bring something like that through, back in the day, they didn't really care. I mean, it wasn't that big of a deal. But you can imagine wheeling that through the airport now. And usually they wanted to look inside, so you'd lift the lid and smoke would come up, and they'd step back and they'd say, okay, go on through. But everybody was so excited about what we were doing. Transporting this sample to Paris for transplantation for a young child. So everybody was very, very excited about this. So the dates were set. Next slide, please. These two are historical documents. These were part of Hal's handwriting. Two of the day planners he had. One of the day planners he had on two different dates. So when Hal passed, we told Beth she didn't have to come down and clean out his office. His administrative assistant, April Maines, and I would do that. So we went through his desk and we went through his filing cabinets. And what I learned after Hal's passing was how much of a hoarder he is. I say this lovingly, how much of a hoarder he was. He kept everything. And we pulled open a drawer, and in that drawer was every desk blotter, calendar that he ever had, a flip calendar that was about this big, and every day planner he ever had. So we're flipping through them. I saw the day that I came in for the interview for that job. And then we found these in 1988, and we flipped them open, and there we were. The one on the left says Scott leaves for Paris. And then that on October 3rd, Hal leaves for Paris, and there's the transplant date. And I don't know if that was the actual transplant date. I know sometimes those things move around, but there's a little bit of history there. So they also decided that it would be cheaper to fly us over and buy a seat for this thing, fly me over, keep me there for a week, and then fly me back, then just fly me over and come back. It was like $2,600 to fly over and come right back, but it was $400 to fly over and back if I would stay there for a week. I said, sure. So my wife went with me, and we had a little mini vacation, but it was big boy that took up 1.5 seats, and then my wife and I shared 1.5 seats, and we took turns eating. But it was a struggle to get that thing up and down the airplane aisle. So the big task at that point was just getting, and so all the plans had been made, the dates were set, we get on the plane, and I was so nervous. I thought, if the plane goes down, we've lost this child's life. all of this effort, all this work is up in smoke. I'm up in smoke. And all I wanted to do was get it in aliens' hands, get it to the hospital, and then my job was done. So when we finally landed in Charles de Gaulle, I sighed this big sigh of relief and knew that all the pressure was off, or so I thought, because then we hailed a cab, got in the cab, and he wasn't French, but he didn't speak any English. And we just conveyed to him that we needed to get this medical sample to the hospital as soon as possible. And that was a mistake, because we took off down the highway, and we were passing cars like they were standing still. And I thought, we've come all this way, and we're just going to burn up in a ball of fire on the side of the road somewhere. And he turned around, one arm on my experiences and stories. I turned around and one arm on the back of the car and found out we were American. Wanted to know if we liked cowboy movies. He loved cowboy movies. John Wayne. He started singing "Happy Trails" to you. "Oh My Darling Clementine," and the whole time, cars are just zipping by. I could feel my wife squeeze my hand tighter and tighter. I look over his shoulder, and we're going 160 kilometers an hour. And I have no idea how fast that was until I got home. But I asked him to slow down a little bit, and he said, sure. So we finally made it to the hospital. We met Agnes Duverger and Ilian, and we handed the sample off, and that was it, and the rest is history. And then it was Hal's turn to take over. And so when Hal went, I don't know whether they kept the sample in the container the whole time until Hal was there or not, but when Hal went, he was going to bring the container back. And he always tells the story that he had to take the container to this hotel room, and it still had nitrogen in it. So he pulled the lid off of it, and he was afraid he was going to asphyxiate. So he slept with the windows open, in late September or early October, and nearly froze to death. But he said it was worth it. So next slide, please. So this was I think this is a picture of young Matthew Farrow and young Elian Gluckman, and you're a cute kid. Next slide, please. Yeah. So just in wrapping up, this is a picture of Zhao Hangok, who did most of the processing for those early cord blood samples, and Hal and me are standing in front of that 15-year cord blood thaw. Next slide. And so in 1993, Hal kind of wanted to start meeting and having some serious talks about this. So we hosted I believe, the first cord blood symposium conference in Indianapolis. There are a couple people you may know and recognize. Bob Good, Ilian, Richard O'Reilly, which I don't know if she told you or not, is that's actually Megan's uncle. Ted Boyce, and some others. Next slide. People you may recognize, much younger versions. Hal, Paolo Rubenstein, John Wagner. Next slide. And it was a good meeting. It was probably half this size, maybe. Next slide. Next slide. And here's you've seen this before, the 20th anniversary of the first cord blood transplant. Next slide. So, in closing, Hal is missed every day. He sacrificed so much of his life for not only cord blood research, but the field of hematology and so many other things, so many other fields. And Megan and Jim are right that he never said no to anyone, and that made my life difficult as his lab manager. He'd want to give somebody space, and he'd say, you need to find some place to put him. Okay. He would give away reagents and supplies, and just to make people successful. He never stopped. As his health declined in the last several months, he continued publishing papers, and he continued to read. And he and I lived about 5 miles apart from each other. And he would have me he didn't come into the lab very often, but he'd have me bring journal articles, and journals, not articles. The actual journals, he read paper. He would not read electronic versions of journal articles. But he wanted me to bring journals, and he would sit there and mark them, and then he wanted me to make Xerox copies and bring them back to him so he could mark them up even further. Because he wanted other people to have access to the journals that he had subscriptions to. But he was blessed with 2 grandkids, 2 wonderful sons, and 2 daughters-in-law, and a wonderful marriage of 50-plus years. And I know Beth misses him terribly, and so does his whole family. But we do too. So thank you.
Thank you, Scott. I'd like to invite Eliane Gluckman and Matt Farrow, who are both here, to come up, and we'll have a panel for the next 10 minutes or so. And then, Eliane, did you want to speak or just talk in the panel? Did you bring slides or no? I don't know. Was that it? Did you bring slides or no? Okay, that's fine. Okay.
We have seen a lot of pictures,
Okay.
so I don't think I need to show that.
Yeah.
Okay.
I think it's better to show.
Okay, great. Then have a seat. Be careful because there are water pitchers on the floor. So for those of us who were there, I mean, this is just a great way to remember how this all started. But for you, all younger people who weren't there, I hope this gave you a real sense of the history. I think it's really important, and one of you said it, to say that if we had to do this again today in our current world, I don't know if it ever would have happened because the regulatory environment's completely different, and it just wouldn't have been so nimble to get the things done that really needed to be done. And Scott, you didn't say it, but Gordon Douglas actually came to Salisbury, North Carolina, where Matthew lives, which is a small town in North Carolina, to be at the delivery. And I think he stayed there a week waiting for Matthew's sister to be born. But there were just a lot of things that happened that show you that when people get together and have a purpose and want to get something done, you know, it can work.
Slides are here. So my slides are here, so maybe I can show them.
Oh, okay, great, great.
It's very few.
You want me to run them for you, and you can stay there?
I just took some.
Right, Eliane's going to
pictures.
do her slides.
I have already,
Because Eliane and Matthew were there when the transplant really happened.
I am really grateful for Al, because he made my career, in fact. I've been doing bone marrow transplant for many years. I did a lot of things, but cord blood is the work which makes my fame. If I did not have cord blood transplant, nobody would know about me. So it's very nice.
I have to say, Eliane is the inventor of reduced intensity transplant, which was a necessary thing for Fanconi because of the DNA repair defect.
So this is why I did this transplant. Maybe we can show the first one. It works. Why? Because I am from Paris, France. As you see, it was difficult for you because you did not speak French. But I can tell you that Americans don't speak French. So it's the same. So I had to learn English, but you did not learn French. Okay. What happened? I have been working on transplant, and I was interested by 2 things. First, I was interested by aplastic anemia, hereditary disorders, and I was the first to describe the toxicity of alkylating agents in Fanconi anemia. And this is the reason why I modified the conditioning regimen. Now everybody knows about it, but at that time nobody knew it. And the results of our transplant with this modified conditioning regimen were much better than the one which used standard protocols. But, I was French, it was never published correctly. And when I sent it to Ash, it was in the trash book. So nobody knew about it. But not everybody, because I was a good friend of Arlene Auerbach, and she knew about our results and our work. So this is how it started. And I was continuing my transplants. They were doing very well. And one day, I was in my office. Hal Broxmeyer, I did not know him before, called me and said, "Are you interested in a cord blood transplant for Fanconi anemia?" I said, "Huh? Interesting, why not?" He explained me the story. I will not enter in details. I was interested for two things. First, I was expert in Fanconi anemia. And second, I had done some fetal liver transplant for aplastic anemia before. It did not work. But I had some knowledge of immature stem cells and development of stem cells. So it fitted well in what I could do. So I was very excited. And I said, "Okay, you come to France. You are welcome. We can organize a transplant." The only thing I wanted was to have Matthew traveling with his family and with the donor, with the sister. In case the cord blood transplant did not engraft, we still had the possibility to collect bone marrow from the daughter. So she was 6 months old. This was possible to collect bone marrow at this young age. So they came, and Ted said how they came. And I asked permission to the ethics committee of our hospital. It was very easy at this time. I sent the thing. They said, "What is different?" I said, "Oh, nothing. It's the same transplant, the same conditioning, the same thing we usually do. The only thing is the difference in the stem cell source, but nothing more." And so they said, "Oh, it's okay if you don't do anything different. Okay, you can go." At that time, it was possible. Now it's not anymore, as you know. So we did it, and I'm glad because it went fine, and Matthew is here, and we have transplanted many other patients with cord blood. Just before coming here, I looked at the Eurocord registry. We have 364 cord blood transplant in our registry, which is a good number, which is very good results. Survival for related cord blood is now 90%, and for the unrelated mismatch is 50%. We are collecting them now. But you see that a lot of progress has been made. So what can I say with that? It was really an adventure, and I will give I will maybe look at, this is the way we collect the cord blood, the way we fold the cord. You don't do that anymore. Right. Not, because it was really it was like big. It was not the best way to do it, but it's a miracle that it didn't spill, that it was not infected, that it did not have a problem at the thawing. But we had luck with us. And the question were, at that time, general applicability, engraftment, we know it's delayed, immunoactivity could be decreased, contamination with maternal cells was a question at that time, and the big banking safety for the donor. And so we did a lot of work to establish cord blood banks. And this is the pioneers. And you can recognize Matthew, Joanne, Arlene Auerbach, myself. This one is a patient who had refractory acute myeloid leukemia in Paris. We had 400 units of cord blood in our bank at that time. We used one of them, and he's still alive and well and never relapsed. Here is Marcela Contreras from UK, and Sashi Gato, I can see, This is Al, John Wagner, Paolo Rebula from Milan, Jan van Rode from Leiden, Peter Wernert, and Pablo Rubinstein. So we were the first to establish the consortium for cord blood banking. And these pictures you have seen with time. And so I am really, really extremely grateful. And what I can say, few words, is that he was a great man. As we say, a mensch. Because he was nice. He was always it was very important for him to support the younger people. He wanted always to make sure that nobody was forbidden or not graded. And he was really a good man. Thank you.
Thank you. So in the last minute, Matthew, and I don't want you to have to do a long speech, okay? I just have a quick question for you. You had no choice well, you had a little choice, actually, and maybe you can tell that part of the story. You had no choice to have Fanconi anemia. You had really no choice in this setup of your mom having, luckily, your sister who was a match and who was healthy. But maybe you could just end by telling us how your family decided, or with you, to go ahead with the transplant. I think you have a microphone that works.
Okay, so yeah, definitely wasn't expected for me to have been born with anything abnormal. My mom and dad actually met hitchhiking. My dad was in the service, and my mom and a friend picked him up, and they've been married 46 years now. And at that time, they were just set out to make a family and didn't know that they were going to be part of something that's going to change the world. My family, they knew something was wrong with me at about 2.5, and we ended up going to Duke University for a little while to do treatments and to figure out what was wrong with me. And they decided that I had Fanconi anemia and I needed a bone marrow transplant. My mother was actually pregnant at the time with my little sister, who is a mother herself now. So was always part of the equation, part of the family. And my father one day set me in our living room and asked me what I wanted to do since it was my body. Did I want to wait till my little sister was older, or did I want to try something that could save other people's lives? And so my dad asked a 4-year-old kid this, and I said I wanted to help other kids like me. And I don't know if I had much of a choice, but it was probably the right answer. And now we're looking at this October 6th will be 34 years post-transplant. And it's just been so honoring to be a part of all this, so honored to be able to have finally met, after 20 years at the 20th anniversary, I met Dr. Broxmeyer and can remember him because I was 5 years old. And he told me a lot of stories, and one of them was one that Scott shared about him almost freezing to death. But he said it was worth it. And that every time that me and Hal got together for certain little events, he would always be one of the most humbling people in the room. And he would treat everyone with kindness and give everyone the time of day to share their stories, share about them, and get to know them. And then he also turned it into a science lesson every time, and which was something that I enjoyed many, many times. And there's just so many fascinating things with this story. Like I know the Curry family, and Emily, the donor for Natalie, we share the same birthday, which is kind of neat. And so there's lots of connections throughout, this family journey. And one of the biggest things I'm grateful for is that Dr. Kurtzberg, Dr. Gluckman and then Dr. Broxmeyer, of course, they give me a chance to live. They give me a chance to have a normal life. They give me a chance to hit milestones that my family never thought would ever happen. You know, being married, having an adopted son, and then also just everything that goes with it, with being divorced and things like that. And then also losing your hair again. So this will be the second time I've been bald. But it's just been a great opportunity, and I'm just very blessed and honored that I was allowed to come and share some stories about our transplant, and just grateful and looking forward to seeing what cord blood has to offer in the future.
Great. Thank you. Thank you. Well, that's a fitting end to the first session. It's time for a break. We'll reconvene here, well, there are two concurrent sessions that will be happening next. So we'll reconvene at 10:30, but there are refreshments in the exhibitor hall. So thank you all so much.